https://pmc.ncbi.nlm.nih.gov/articles/pmc 7486059 potent antiviral effect of silver nanoparticles on SARS-CoV-2 Rajesh Jeremiah A. K. Miyakawa A. Takashi Morita A. Yutaro Yamaoka A.B. Akihide Ryo A. Operator author information article notes copyright and license information PMCID PMC 7486059 PMID 329582550 Abstract The pandemic of COVID-19 is spreading unchecked due to the lack of effective antiviral measures. Silver nanoparticles AgNP have been studied to possess antiviral properties and are presumed to inhibit SARS-CoV-2. Due to the need for an effective agent against SARS-CoV-2, we evaluated the antiviral effect of AgNPs. We evaluated a plethora of AgNPs of different sizes and concentration and observed that particles of diameter around 10 nanometers were effective in inhibiting extracellular SARS-CoV-2 at concentrations ranging between 1 and 10 ppm, while cytotoxic effect was observed at concentrations of 20 ppm and above. Luciferase-based pseudovirus entry assay revealed that AgNPs potently inhibited viral entry step via disrupting viral integrity. These results indicate that AgNPs are highly potent microbicides against SARS-CoV-2 but should be used with caution due to their cytotoxic effects and their potential to derange environmental ecosystems when improperly disposed. Keywords: silver nanoparticles, colloidal silver, SARS-CoV-2, COVID-19 highlights silver nanoparticles inhibit extracellular SARS-CoV-2. 2 to 15 nanometer size particles showed robust inhibition of SARS-CoV-2. SARS-CoV-2 was potently inhibited at concentrations above 1 ppm. Naked silver nanoparticles exhibited cytotoxicity from 20 ppm onwards. 1. Introduction: the elemental metal, silver Ag, has broad-spectrum antimicrobial action against various bacteria, fungi, and viruses. Due to their versatility, Ag nanoparticles, AgNP, have currently found their way as a microbicide for biological surfaces in various forms such as wound dressings, medical devices, deodorant sprays and fabrics. Several studies have demonstrated the potent antiviral action of AgNPs against various human pathogenic viruses such as respiratory syncytial virus, RSV, influenza virus, norovirus, hepatitis B virus, HBV, and human immunodeficiency virus, HIV, 1. In addition to these viruses, since Ag has been demonstrated to kill SARS-CoV, we hypothesized the strong possibility of AgNPs to inhibit SARS-CoV-2, 3. Till date there are no studies directly demonstrating the effect of AgNPs on SARS-CoV-2. We tested colloidal silver, CAG, plain elemental Ag nanoparticles of different diameters, AgNPN, and polyvinylpyrrolidone capped 10 nanometers silver nanoparticles, PVP AgNP10, against SARS-CoV-2 to find the most effective size and concentration of Ag that could inhibit SARS-CoV-2. We propose that AgNPs could be used on inanimate and non-biological surfaces to efficiently control the ongoing COVID-19 pandemic while simultaneously exercising care not to abuse it. 2. Materials and methods 2.1. Cell culture and virus propagation VAROE6/TMPRSS2, VAROE6 cells stably expressing the transmembrane serine protease TMPRSS2, JCRB number 1819. And CALO3 cell lines were cultured in DMEM containing 10% FBS, 4. SARS-CoV-2, JPN/TY/WK521, was obtained from NIID, Japan, stored in aliquots at -80 degrees Celsius and handled in biosafety level 3, BSL-3. While performing SARS-CoV-2 infection studies, DMEM containing 2% FBS was used. 2.2. Plaque assay was performed SARS-CoV-2 on VAROE6/TMPRSS2 and CALO3 cell lines as described elsewhere with minor modifications. 5. 96 well plates were seeded with 5/104 cells per well in 10% FBS/DMEM and allowed to grow overnight. Viral solution was diluted in 10-fold serial dilutions in 2% FBS/DMEM. The supernatants from wells were removed and replaced with viral dilutions in correspondingly labeled wells and incubated at 37 degrees Celsius for 96H, after which the cells were fixed with 4% formalin and stained with 0.25% crystal violet to visualize the plaques against a white background. Median tissue culture infecting dose, TCID 50, and multiplicity of infection, MOI, were calculated from quadruplicate tests. 2.3 Silver formulations PVP AgNP 10 at 20 ppm stock concentration, CAT No. 795925, and CAT No. 85131, were obtained from Sigma. AgNPs of different sizes, AgNP2, CAT No. US 7150, AgNP 15, CAT No. US 7091, AgNP 50, AgNP 80, and AgNP 100. US 1038W, were purchased from US Research Nanomaterials Incorporated. All of the silver formulations were dispersed in water and the desired concentration was prepared by diluting in sterile distilled water. 2.4 Cell Viability Assay. The Cell Titer Glow Cell Viability Assay, PROMEGA, is a luminescence-based assay which quantitatively detects live cells based on adenosine triphosphate, ATP, levels. Cell death subsequent to Ag-mediated cytotoxicity or viral infection could be rapidly quantified using cell titer glow. 6. 50 μl of cell titer glow substrate, PROMEGA, was added to the cells and their viability was measured based on the luminescence intensities detected by GlowMax Discover system, PROMEGA, 10 minutes later. 2.5 RT-qPCR viral RNA was extracted from culture supernatants using QIAMP viral RNA mini kit, Chagan, and stored at -80 degrees Celsius until further analysis. The extracted viral RNA was quantified using CFX96 real-time system, Biorad, with a TaqMan fast virus one-step master mix, Thermo. Using 5-AATTTTGGGGACCAGGAC3 foot and 5-TGGCAGCTGGTTAGGTCA3 as the primer set and 5-FAM-ATGTCGGCATTGGABHQ3 as probe. 7. 2.6 Cytotoxicity studies. CAG or AgNPs at desired concentration were added to VEROE6/TMPRSS2 or CALA3 cell lines grown in 96 well white plates and were incubated at 37 degrees Celsius for 48 or 96H respectively, after which the cells were washed with PBS and the viability was quantified using the cell titer glow assay. 3. Silver and SARS-CoV-2 interaction studies. 3.1 Virus pretreatment assay. Virus at a MOI of 0.05 for VEROE6/TMPRSS2 or 0.5 for CALA3 in DMEM containing 2% FBS was incubated with 2 ppm solution of AgNP for 1H at 37 degrees Celsius. The corresponding virus AgNP mixture was added to VEROE6/TMPRSS2 or CALA3 cells in 96 well plates and incubated for 48H or 96H respectively at 37 degrees Celsius. Cell viability was quantified by cell titer glow assay and the viral copies in supernatant were quantified by RT-qPCR. 3.2 Cell post-treatment assay. VEROE6/TMPRSS2 cells were infected with SARS-CoV-2, MOI equals 0.05, and incubated for 2H at 37 degrees Celsius. The wells were washed with PBS to remove extracellular viruses and topped up with DMEM containing 2% FBS with 2 ppm PVP AgNP10 and incubated for 48H at 37 degrees Celsius. Cell viability was quantified by cell titer glow assay and the viral copies in supernatant were quantified by RT-qPCR. 3.3 Cell pretreatment assay. VEROE6/TMPRSS2 cells were treated with 2 ppm PVP AgNP10 and incubated for 3H at 37 degrees Celsius. The wells were then washed with PBS to remove free AgNPs in medium and topped up with DMEM containing 2% FBS with SARS-CoV-2, MOI equals 0.05, and incubated for 48H at 37 degrees Celsius. Cell viability was quantified by cell titer glow assay, and the viral copies in supernatant were quantified by RT-qPCR. 3.4. Immunofluorescence cells were fixed with 4% paraformaldehyde and permeabilized with 0.5% Triton X100, and then were blocked with blocking 1-nacali at room temperature for 15 minutes. The cells were incubated with the polyclonal antibody against SARS-CoV-2 nucleocapsid protein antibody, 1:100 dilution, Novus NB 100-56576, at room temperature for 1H. After incubation, cells were stained with Alexa 568 labeled anti-rabbit antibody, 1:1000 dilution, thermo, for 1H at room temperature. The nucleus was stained with prolonged gold anti-fade mount with DOPI, thermo. The images were taken by fluorescence microscope BZ9000, Kians. 3.5. Pseudovirus entry assay pseudotype lentivirus was produced by transient transfection of HEK293 cells with PNL4-3. Luke.re- and PSARS2 spike flag at a ratio of 1:1. Culture supernatants containing pseudoviruses were collected 48H after transfection and filtered through a 0.45 micrometers mil-XHV filter, Merck. For the entry assay, VAROE6/TMPRSS2 cells seated in 96 well plates were washed and inoculated with 100 ml of medium containing pseudovirus with or without PVP AgNP10. Neutralizing antibody was used as a control for entry inhibition. 48H after inoculation, cells were washed and added with 40 ml of bright glow substrate, Promega. Luciferase activity is measured with GlowMax Discover System, Promega. 4. Results 4.1. Cytotoxicity, effective dose, and contact time. AgNPs exhibit cytotoxicity to mammalian cells by generating reactive oxygen species, ROS 8. We wanted to assess the concentration-dependent cytotoxicity exhibited by Ag on the cell lines VAROE6/TMPRSS2, non-human origin, and CALO3, human lung epithelial cell. CAG was serially diluted and added to the cells in 96 well plates, and the cell viability was assessed after 48 hours using cell titer glow assay. Ag was found to exhibit cytotoxicity at concentrations from 20 parts per million, ppm, onward in both VAROE6/TMPRSS2, Figure 1a, and CALO3 cell lines, Figure 1b. Figure 1. Figure 1 open in a new tab. Cytotoxicity of colloidal silver on mammalian cells. 1a, cytotoxicity exhibited by serial concentrations of colloidal silver on VAROE6/TMPRSS2 cells. 1b, cytotoxicity exhibited by serial concentrations of colloidal silver on CALO3 cells. Since CAG contains particle sizes varying from 1 to 1000 nanometers, we used it as an initial screen to ascertain the effect of AgNPs on SARS-CoV-2. The multiplicity of infection, MOI, of SARS-CoV-2 was calculated by independent experiments and was found to be 0.05 and 0.5 for VAROE6/TMPRSS2 and CALO3 respectively. Viral suspension at a fixed MOI was treated with each serial dilution of CAG for 1H and then inoculated to VAROE6/TMPRSS2 and CALO3 cells. Cell viability of infected VAROE6/TMPRSS2 was assessed after 48 hours to identify the proportion of cells killed by the virus, and the viral load was quantified in CALO3 cells using real-time reverse transcriptase quantitative polymerase chain reaction, RT-qPCR. After 96 hours, CAG showed robust antiviral effect denoted by increased viability of infected VAROE6/TMPRSS2 cells at concentrations ranging between 1 and 10 ppm, Figure 2a. In CALO3 cells, significant viral load reduction was observed at similar CAG concentration range, Figure 2b. While metal ions are known inhibitors of PCR at high concentrations, we confirmed that at the effective concentration, 2 ppm, Ag did not inhibit amplification and is suitable to analyze viral RNA in Ag-containing samples, Fig. S1, 9. Since 2 ppm was also 10-fold lower than the cytotoxic concentration, it was chosen as the desired concentration for further testing. Figure 2. Fig. 2. Open in a new tab. Concentration and dose-dependent antiviral effect of naked silver nanoparticles on SARS-CoV-2.2a. Colloidal silver rescues VEROE6/TMPRSS2 cells from SARS-CoV-2-mediated cell death in a concentration-dependent manner. Error bars obtained from triplicate testing. P-value less than or equal to 0.005, *operator**operator**. 2b. Concentration-dependent inhibition of SARS-CoV-2 replication in CALO3 cells by colloidal silver. Error bars obtained from triplicate testing. P-value less than or equal to 0.001, *operator**operator**. 2c. Silver nanoparticles exhibit size-dependent antiviral action against SARS-CoV-2 in VERO/TMPRSS2 cells. Error bars obtained from triplicate testing. P-value less than or equal to 0.005, *operator**operator**. 2d. Size-dependent viral inhibition of SARS-CoV-2 by silver nanoparticles in CALO3 cells. Error bars obtained from triplicate testing. P-value less than or equal to 0.001, *operator**operator**. Previous studies have documented the size-dependent potency of AgNPs in viral inactivation, with less than or equal to 10 nanometers particle size reported to have maximum antiviral effect, 10. Since CAG contains varying sizes of Ag particles, we predicted that the particles around 10 nanometers size in CAG would have exerted the antiviral effect. To prove this, we employed the virus pretreatment assay, VPRA, to test antiviral effect of AgNPs of different sizes ranging from 2 to 100 nanometers on extracellular viruses. Virus was treated with 2 ppm solution of AgNPs of different sizes for 1H, and the virus AgNP mixture was added to VEROE6/TMPRSS2 and CALO3 cells. The cell viability was quantified by cell titer glow assay in VEROE6/TMPRSS2, and the viral copies in supernatant were quantified by RT-qPCR in CALO3 cells. Antiviral effect was noted with AgNPs of size ranging from 2 to 15 nanometers, figure 2c and d. AgNP2 showed cytotoxicity at 2 ppm while other sizes did not, fig. S2. Hence we chose PVP AgNP10 for further testing. Since we observed excellent antiviral activity in VPRA at 1H, we wanted to know the minimum contact time required by Ag for viral inhibition. Time course study performed based on VPRA with PVP AgNP10 revealed significant inhibition beyond 30 minutes of contact, fig. S3. 4.2. Silver inhibits extracellular viruses by preventing viral entry. We next performed the VPRA, cell post-treatment assay, CPA, and the cell pretreatment assay, CPRA. On SARS-CoV-2 using PVP AgNP10 in VEROE6/TMPRSS2 to observe the effect of Ag on extracellular and intracellular viruses, figure 3a. VPRA showed effective inhibition of extracellular free virions characterized by both the reduction of cell death and also a steep fall in the viral load to negligible levels, figure 3b and c. We further performed the CPA to detect the ability of Ag to suppress virus in already infected cells. In this experimental design, VEROE6/TMPRSS2 cells were allowed to be infected with SARS-CoV-2, MOI 0.05, for 2 hours, after which the extracellular viruses were washed and then the infected cells were treated with 2 ppm of PVP AgNP10. We observed significant protection of infected cells and suppression of viral load with PVP AgNP10, figure 3b and c. Additionally, we performed the CPRA to assess the ability of silver pretreated cells to resist viral infection. VEROE6/TMPRSS2 cells were incubated with 2 ppm PVP AgNP10 for 3H, after which the cells were washed to remove unbound silver followed by infection with SARS-CoV-2, MOI 0.05. At the end of 48H, the virus was found only to be partially inhibited, figure 3c. Figure 3. Figure 3. Open in a new tab. Silver nanoparticles effectively inhibit extracellular SARS-CoV-2.3a, Schematic representation of virus pretreatment assay, top panel, cell post-treatment assay, central panel, and cell pretreatment assay, bottom panel. 3b. Performance of PVP-coated 10 nm silver nanoparticles in the 3 study designs with respect to rescue of cells from SARS-CoV-2 infection. Error bars obtained from triplicate testing. P-value less than or equal to 0.005, *operator**operator**operator. 2c. Performance of PVP-coated 10 nm silver nanoparticles in the 3 study designs with respect to reduction of SARS-CoV-2 replication. Error bars obtained from triplicate testing. P-value less than or equal to 0.001, *operator**operator**operator. We confirmed the size-dependent antiviral effect of PVP-AgNP10 using the immunofluorescence analysis carried on VPRA experimental model. SARS-CoV-2 infection was effectively averted by AgNP10 and not by AgNP100, Figure 4a. The plaque assay revealed that silver achieved complete inhibition of 0.05 MOI, which is 1 log 10-fold more than virus control. Partial inhibition was observed with higher viral loads starting from 0.5 MOI, Figure 4b. To assess the role of AgNPs in viral entry, we performed the Luciferase-based pseudovirus entry assay. PVP-AgNP10 potently inhibited pseudoviral entry characterized by significant reduction of luciferase activity similar to that of the neutralizing antibody used as control, Figure 4c. Figure 4. Figure 4 open in a new tab. Characteristics of PVP-coated 10 nm silver nanoparticles in SARS-CoV-2 infection. 4a. Immunofluorescence imaging comparing the effect of 10 nm and 100 nm silver nanoparticles against SARS-CoV-2 infection in VAROE6/T-MPRSS2 cells. Cell nuclei, blue, and SARS-CoV-2 nucleocapsid protein in cytoplasm, red. NC negative control. 4b. PVP-coated 10 nm silver nanoparticles protect VAROE6/T-MPRSS2 cells from SARS-CoV-2 infection-mediated cell death. Crystal violet staining reveals partial protection with visible plaques, red arrowheads, and complete protection with absence of plaques, black arrowheads. 4c. Pseudovirus entry assay. PVP-coated 10 nm silver nanoparticles inhibit entry of pseudovirus in VAROE6/T-MPRSS2 cells. NC negative control, NAB neutralizing antibody. For interpretation of the references to color in this figure legend, the reader is referred to the web version of this article. 5. Discussion. Ag is long known for its antimicrobial effect, and the antiviral property of AgNPs is being extensively researched with renewed interest in the recent past. 1. The exact mechanism by which AgNPs exert its killing effect on viruses is still obscure. However, it has been consistently observed that AgNPs interact with the structural proteins on the surface of extracellular viruses to inhibit infection in the early phase, by either preventing viral attachment or entry, or by damaging the surface proteins to affect the structural integrity of virions. 11-12. In the current study, we have obtained similar findings in the VPRA where AgNPs effectively inhibits extracellular SARS-CoV-2 to protect the target cells from infection, and the pseudovirus entry assay revealed that AgNPs interfere with viral entry. AgNPs have been shown to preferentially bind to viral surface proteins rich in sulfhydryl groups and cleave the disulfide bonds to destabilize the protein, thereby affecting viral infectivity. 11-13. Studies on HIV have shown that AgNPs associate to the disulfide bonds that are in close proximity to the CD4 binding domain of the GP120 surface protein. 11. Hadi and Bhattacharya have demonstrated the importance disulfide bonds in binding of SARS-CoV-2 spike protein with the angiotensin-converting enzyme 2, ACE2, receptor and the disruption of which lead to impaired viral binding to the receptor. 14. Considering the mechanism of action of AgNPs shown by other authors, it can be presumed that AgNPs exert their antiviral effect on SARS-CoV-2 by disrupting the disulfide bonds on the spike protein and ACE2 receptors. Further studies are being conducted to find the antiviral mechanism of AgNPs on SARS-CoV-2 and elucidated in detail subsequently. AgNPs have also been claimed to possess intracellular antiviral action by interacting with viral nucleic acids. 15. We observed a partial antiviral effect in CPRA, as there was some amount of reduction in the viral load in cells pretreated with PVP-AgNP10. While the reason for this effect is not known at present, it is possibly explained to be either due to the destruction of disulfide bridges on ACE2 receptor or due to a true intracellular mechanism, thereby inhibiting serial viral infection of newly produced virus from infected cells to uninfected cells. Also, since Ag binds nonspecifically to proteins, their use as antiviral agents might also cause some cellular dysfunction. Further studies are required to more precisely explain the holistic effect of Ag in vivo. Several studies have reiterated the size-dependent antiviral effect of AgNPs, with particles around 10 nanometers diameter being most effective. 1. This has been attributed the higher stability of interaction to the viral protein achieved by 10 nanometers particles, which is not capable by larger particles. 11. Consistent with this, we also observed anti-SARS-CoV-2 activity only with AgNPs of diameters ranging from 2 to 15 nanometers. Our immunofluorescence study corroborated the above phenomenon, as we observed that PVP-AgNP10 completely inhibited SARS-CoV-2 but AgNP100 did not. AgNPs can be generated by several methods and can contain reducing agents and capping agents along with the metal particles. 16. Coated or capped AgNPs are found to be more advantageous than plain AgNPs, as coating increases stability, decreases agglomeration, and reduces cytotoxicity of AgNPs. 17. Among the coated AgNPs, PVP-capped nanoparticles are widely studied for biological use.